Okay, so what cellular transport process is most responsible for transporting water up the stem? Osmosis. Okay, where in the stem would you find the phloem? Nope. It has to be just osmosis because cohesion and adhesion are not cellular transport methods. Just inside of the bark, on the outside of the stem. I'll accept anything that would imply it's the kind of the outside, just inside the outside layer, right? Okay, so near outside, just under bark, okay, any of those kind of things is fine. Okay, what gases would be stored in the spongy layer? CO2 and oxygen. CO2 and oxygen. Nitrogen is always, it's not stored there, right? These two gases are exchanged, okay? So we'll say CO2 and oxygen. We'll make that one out of 2 as well, so it'll be out of 12. Okay, number 4, what was the major advantage of multicellularity? Allowed organisms to get bigger. Simple as that. Okay, allowed organisms to get bigger. If you wrote something about overcoming surface area to volume problems, that's why they could get bigger, so that's also right. Well, specialization is what had to happen to become multicellular. The advantage of multicellularity was size. Yeah, the main advantage was size. Okay. No. Okay. Multicellularity, guys. That was the whole message that cube lab that was due yesterday was about. Being multicellular means you don't have to worry about diffusion not working over large distances. Okay. All right. So we'll just write in one word the answer is kind of size there. Okay, number 5. What is the difference between a true multicellular organism and a colony of independent cells? It's the one you wanted to say last time. Specialization. Okay, that is the difference. In a colony, all the cells are the same. In a true multicellular organism, the cells are different. Some are specialized to do certain jobs. Okay, what cells in the plant change their shape the most by changing the amount of water in their vacuoles? Guard cells. Okay, I won't accept stomates on that one because guard cells make up the stomates. It specifically asked for the cells. The structure the cells make up is the stomates. Okay, I'll let it go today, but in the future, okay? If it asks specifically about which cells, the guard cells make up the stomate. The stomate is not a cell in and of itself, okay? That's fine. Yeah, just. Okay, the tubes that conduct water up the tree are called xylem. Okay, what are the 2 functions of the stem? Support and transport, or conduction and support was what was in your notes, okay? That one's out of 2 as well. As days get shorter, a plant would secrete what to signal dormancy? Abscisic acid. I spelt it wrong. No, I didn't. Abscisic. Yes, I did. Abscisic acid. Okay, that's not out of 2 marks. They either have abscisic or they don't. Okay? Could you tell they meant abscisic? Okay, then it's fine. If it starts with a Z, then it's not right.
It's okay, we spelled it wrong too.
Yeah. Okay, and number 10. What substance is secreted by plants as a growth hormone lengthening the plant? Auxins. Okay, out of 12. Give him a mark out of 12. Let him see it, and then I would like them handed in right in front of Brendan here. No, I made it out of 12 because I made this one out of 2, this one out of 2. Okay, so what we're working on today, guys, is an activity that's going to review essentially 2 things. The first thing is going to review the factors that affect plant growth and a little bit of the stuff on plant growth and development from yesterday. Okay? The second thing that it's going to review is the scientific method. That is variables, designing a procedure, coming up with a hypothesis, all of that kind of stuff. Because your task today in class, okay, this must be done by the end of scheduled help. Okay, it is not homework. I'm not doing this to give you extra homework. It's an activity to review some things. Okay, by that time, what I want to see is an experiment designed from the problem to the procedure, okay, including the procedure. Right, an experiment that will test the effect of a variable of your choice on the growth of a plant. Okay, so you've got to come up with the problem. The problem is fairly straightforward. Once you've picked what you want to test the effect of, that's your problem. What is the effect of this on the growth of a plant? Okay, that's really kind of the easiest part. Then you'll have your variables. Okay, the thing that you're wanting to test the effect of is going to be which variable? Like, let's say I want to test the effect of, let's say, different types of soda pop on the growth of a plant. But not that anyone would do that. That's why I'm picking it. Okay? So I'm going to have an experiment where I feed one Coke, I feed one Pepsi, I feed one root beer, I feed one Orange Crush, Grape Crush, okay? Whatever. I just give it all different kinds of soda. Okay? And then I probably have one plant that just gets water, so I have a comparison. Okay? I'm testing the effect of different fluids, essentially. Which variable is that? That's the manipulated variable. Okay? Now, I know it looks like I'm testing more than one thing. Am I? No. All I'm testing is the effect of different liquids on the growth of a plant. Okay? I can test different liquids as long as I don't test anything else but the liquids. All right? Now, responding variable for everybody is going to be the same thing. What am I watching for? How much it grows. Now, are there different ways to measure that? You should probably include some qualitative and quantitative ways that you're going to measure that. Okay, quick review. Qualitative ways would be things that you would write down that you're not actually measuring, like how does the plant look? Okay, is the plant discolored? Is the plant look stunted? Okay, are the leaves misshapen? Things like that. Okay, quantitative things are I measured how tall it was in centimeters. Okay, or I took the whole plant and its pot and took its mass every day. Okay, those are quantitative measures. They're actual numbers and measurements, okay, of the growth of the plant. Does everyone follow? Okay, so everybody's responding variable is going to be the growth of a plant. You want to decide and explain how you're going to measure that. Controlled variables. This is the most important part for you. Okay, you need to mention 3 controlled variables. Okay, 3 things that you've got to make sure are going to stay the same throughout your entire experiment. Okay, these are things that could affect the growth of a plant. Everybody follow? Things that are not what you're manipulating, but things that you know plants need. Okay, now, you might mention these in your variables, but here's where I'm checking them. I'm checking them in your procedure. If you design an experiment where you've said, I'm controlling these 3 things, and then in your procedure, you say something different, okay, then that's going to affect your mark. All right? So things to consider. If I've got a whole bunch of plants and I'm testing them, I probably would put them, if it was me, I'd put them all on that windowsill. Okay? As long as I wasn't testing different kinds of light. That would be fine, because I'd be controlling a lot of other things about them. Okay? Like, they're all in this room. So everything in this room is staying the same for all of them. Okay? Those are the kinds of things you need to consider without me giving them all away, because part of this is seeing whether you can figure that out or not. All right? You have to come up with a hypothesis. Okay? If and then statement that says, if this affects plants in this way, and then I do an experiment that looks like this, then these are going to be my results. Okay? You tell me what are the results going to look like. Right? And then you're going to have a list of materials. Okay, so essentially you're putting a lab together here. Okay, you look at, you know, kind of the order a lab goes in. Okay, problem, variables, hypothesis, materials. Okay, and then a procedure. And in that procedure, lay it right out. Okay, explain how you're setting everything up. How big a pot do you need? How much soil do you need? Okay, those are things you can kind of take guesses at. But to give you some idea here, okay, this is a 5-liter pot. Okay, it holds 5 liters of soil. All right. You can choose which plants you want to test. I would say that might be something you'd want to make sure you're not testing like 4 different kinds of plants. That might affect your results negatively. Okay, it'd be hard to compare carrots to pine trees. Okay. So things like that. Those are all things to consider. Everybody kind of follow what we're doing? Okay. The last thing that's going to be different. Okay, so everything else has been the same as an experiment we've done up to this point. The thing that's going to be different is a short paragraph right at the very start, before even the problem. Okay, this short paragraph is called the abstract. In real science, you have to sell your research to somebody. Okay, that is, if you're going to have money to do your research, you have to go in and have somebody essentially sponsor what you're doing. Okay, unfortunately, that means they own your results. All right, so if you come up with the cure for cancer, okay, in research, some big pharmaceutical company owns that. Okay, not you. Right, you get paid by them to develop it, and then you get a warm feeling inside for solving the biggest health crisis on planet Earth, and they get filthy rich. Okay, that's unfortunately how science works. You do not meet a lot of filthy rich scientists. Okay, it's not a get-rich profession. All right, everybody follow me on that one? Okay, so what you're doing in an abstract is a sales pitch. Okay, in your abstract, you're going to tell me the following things. Here's what I know about plant growth. This is what I want to test, and here's a possible application of that in the real world. Okay, so it's like 3 things get covered in there. Okay, here's what I know about plant growth. This is what I want to test, and here's an application of it in the real world. Okay, that's what goes in your abstract. So it's a short paragraph. Like, how could it be used? Like, if I'm selling it to a company, they don't want to invest in my research if all I'm going to get out of it is, hey, plants grow good in the light. They're like, hey, stupid, we already know that, right? Okay, that's not going to make us any money. How is your thing going to make us money later? Okay, so it's a sales pitch. Yeah? Okay, does everyone follow what the expectations are here? All right, so when this gets handed in, it's going to look like a normal lab report. Problem, okay, abstract, problem, variables, hypothesis, materials, procedure. Okay, you can't go any further than procedure because you're not actually doing this experiment. There's no analysis. Okay, you don't have any data to analyze, so there's no analysis, no conclusion, no evaluation. All right, questions? No, this is, I don't want this to be for homework. You have enough stuff due for me already. Okay, this is just something to help us review some things in class today. You can. You have to. Okay? Yes, you have to have this done. I made the due date 11:56. Okay. All right, let's get to it. If you got questions while you're working there, let me know.